刘院士，下午好哈。那你好，那因为我们今天当初也是说我们就是全程用英文，所以我就 switch 到那个 English 的部分哈。OK， it's our great honor to invite the Fudong Liu academia to have the talk today. And because everybody know right now is the COVID-19 outbreak, so we don't want to block any possibility for the master symposium. So today we invite Professor Fudong Liu give us a nice talk. And let me introduce the Fudong Liu a little bit. He is a chemist, but also is a medical doctor, and he focused on the glycoprotein research maybe 20 years, and have a lot of the outstanding finding. And Professor Liu, as the academician after the 2012 in Academic Seneca. And he also, as I know, you are the president of the immunology societies right now. Yeah.
Oh, thank you. I just finished my term.
Finish your term.
Right.
Okay.
So the new president is a Situ.
Oh, okay. Because next year is the physiology society in charge for the biomedical conference. So I read some information as I know. Okay. So today's we have like a 1 hour's talk. Let the Professor Liu to give us about the Glycan-binding protein from the biology and therapeutic potential. So give the warm hands to welcome our Professor Liu.
Thank you. I want to thank Tzu University College of Medicine for this very kind introduction, and allow me to share with you my knowledge in glycobiology and share with you my research. So it's unfortunate because of the COVID-19 situation, I'm not able to visit in person. I hope when the pandemic is better controlled, I visit the campus again. So my topic is glycoscience. So 2012, less than 10 years ago, or almost 10 years ago, the National Research Council of National Academies, this is the United States, coordinated jointly by National Academy of Sciences, NAS, National Academy of Medicine, or maybe also with engineers. They jointly prepare a document entitled Transforming Glycoscience, a roadmap for the future. And they state that it means if we transform glycoscience according to the roadmap proposed, it can make contributions toward understanding and improving human health, as well as many other benefits, including, I remember that even generation of new materials. And they emphasize that now, meaning that at that time, is the time for glycoscience to be embraced, these keywords embraced broadly by the research community. And they say that although these challenges are substantial and complex, and you might get the impression that indeed the area I'm going to talk about is a little bit complex. But the results of achieving these goals have the potential to impact science in exciting ways. Then just merely about 5 years later, meaning about 3-4 years ago, Nature, there's an article published in Nature, Sweet Success, and it's highlighted that biologists are diving into sugar molecule research thanks to new tools and techniques. Those new tools and techniques are glycomics tools and techniques. And by glycomics, this is just a very nice photo or diagram of what glycobiology is about. We have glycoproteins and include O-linked glycans and N-linked glycans. Also glycolipid and glycosaminoglycans, proteoglycans. Inside the cell, there's a very important glycosylation. That is O-linked N-acetylglucosamine. This is just monosaccharide. Hundreds or even a large number of intracellular proteins are glycosylated this way. Just like many are phosphorylated. In fact, the site for phosphorylation is this phosphorylation on serine and threonine, not on tyrosine. Serine, threonine phosphorylation and the O glycosylation interchangeable, meaning that the site that can be phosphorylated can also be glycosylated. They can go back and forth. But I will not talk about this glycosylation today. And also like to mention that for the purpose of today's presentation, and also for you to, when you get encounter in this area, just think about this glycan as a pattern or as some kind of shape. Just like when you look at proteins, you don't necessarily think about amino acid sequence. You think about maybe one kind of shape, and there's a binding site that bind ligand. Same thing. So I know that many of you might not be familiar with glycosylation, but these glycans, actually, when you think about it, you don't think about complex structure. You think about this as a pattern, especially for today's presentation. So I mentioned that many of the tools and techniques are developed and making actually this sweet success. Glycomics is the systematic study of all glycan structure of given cell type or organism. However, just like when we talk about proteomics, we learn about we determine the structure of proteins. We do not necessarily know the function unless the protein is homologous to some known protein with a known function. Same thing for glycomics. So when you determine the glycan structure, you don't necessarily know the function. So there's an area called the functional glycomics. So the authors stated that to decipher the function, we need to identify proteins that recognize the glycans that can translate information encoded by a specific glycan structure. So the functional glycomics was named to define the concerted and regulated action of carbohydrate binding protein or glycan binding protein, which is topic of today's talk. I don't quite completely agree with this definition because I think glycans, they have other functions in addition to being recognized by glycan binding proteins. For example, they can keep the protein, the glycoprotein, in certain shape, certain the stability, and certain conformation. However, I think this is a very convenient term to talk about how we move from glycomics to functional glycomics, and that is the study of glycan binding protein. Indeed, this is the very major branch of glycobiology. In fact, it's also one direction with less barrier in terms of getting into glycoscience by studying these glycan-binding proteins. However, I mentioned today's at the interview in the morning that after today's talk, by no means I wanted everyone to jump into glycoscience. You have your professor have your research topic, and the student have their thesis or their learning, still learning science, and but these days it's the way research is being conducted. Usually, we go through high throughput screening or non-biased survey to get the overview of what's going on in the system you're interested or the disease you're interested in studying. Then, if through that exercise there are some glycan binding proteins that surfaced, then I would like you to embrace that opportunity and don't just say that, well, this is actually opportunity to get into glycoscience. In fact, I got into glycoscience totally by accident, but I won't go into that today. So these are major types of glycan binding proteins. Glycan binding protein, another term is old term is lectins. So lectin in Chinese is Ningji Su. So glycan binding proteins are lectins. They are, I think you are familiar with many different lectins, especially plant lectins. So I'm sure you're familiar with selectin. There are 3 selectins: L-selectin, E-selectin, and P-selectin. These are very famous glycan binding proteins. And these are C-type lectins. C stands for calcium. So these are the activity require calcium, the presence of calcium. Then there are hundreds or even maybe over 1,000 C-type lectin-like receptor or C-type lectin. Put the hepatic lectin here. I won't get into this very large family proteins. Then another interesting and important family is the Sig-like or I-type. So I will talk about this later. And galactans, which is the major topic of today's presentation, different from other lectins in that they are not transmembrane proteins, and they do not have so-called leader sequence or signal sequence. So they are not necessarily made to be secreted. So they are actually reside inside the cell, although you can find that outside the cell. This make the study of galactans a little bit challenging because they are, as you see, the glycoligand recognized by all these lectins reside outside the cell, on the cell surface and extracellular matrices. Although they are made inside the cell, right? But they are expressed outside. And galactans, glycan binding protein, but they reside mostly inside the cell. I will dissect that, what they do later. So I want to share with you challenges associated with investigation of glycan binding proteins. So I mentioned that from proteomics to protein function require our understanding of what the protein bind to or interact with. So take cytokines, for example. Each cytokine has its unique receptor. So many of cytokines you're familiar with, or we call interleukins, or even some mediators like hormone, they have their unique receptors. The glycan binding proteins are different. So when we go from glycomics, we know to functional glycomics, we go from we know the glycan structure to its function. There we definitely can connect to glycan binding proteins, but given glycan binding proteins may engage different glycoproteins on the cell surface because different glycoproteins may express the same glycan ligands. So that is glycan binding protein bind glycans. The glycan can be carried by many different glycoproteins. So when you engage that glycan, maybe many glycoproteins are engaged. And also, when glycan-binding protein get outside the cell, you actually might not be able to predict what glycoprotein you will bind to, because there are many possibilities for them. So I will talk about selectins, which you more familiar with, and then Siglex, and then spend about the second half of my talk on galactans. There, I talk about extracellular function first, and actually to remember, I have question mark here, and then please, I hope I can explain clearly why this question mark. Actually, this has to do with the previous slide, that is a challenge associated with this field of research. Then I will spend most of the time on intracellular function. There, I mentioned that inside the glycoligands are outside the cell, but so inside the cell, actually galactans can function in glycan-independent fashion. And I will share with you our study on galactan-3 and disease and galactan-7 as a relationship to psoriasis. But then share with you, I believe, very exciting development in the field of galactan, in fact, a field of glycobiology. That is, galactans may be unique in this aspect. That is, they have glycan-dependent function inside the cell, inside the cell, intracellularly. So selectin, as I mentioned, that this is a C-type lectin. C stands for calcium. It's calcium dependent. And the L-selectin, E-selectin, and P-selectin. This is almost among the first family of glycan-binding protein that were discovered, and they actually enhance the appreciation among the scientific community of the importance of carbohydrate binding. And these are themselves glycoproteins, but these are lectins, but they're themselves glycoprotein. Lectins are not necessary glycoproteins, right? But these three selectins happen to be glycoprotein as well, and they bind to carbohydrate presented on glycoconjugates, and they are very well known for being cell adhesion molecules. They mediated cell adhesion cascade, and during inflammation, selectin serve as these biomolecules that are responsible for trafficking of leukocytes from their origin to the site of inflammation. So this is already a very classical diagram, and this is pretty much like a textbook, something you will read in standard textbook. So P-selectin and E-selectin, P actually stand for platelet, but they also expressed by endothelial cells. E stand for endothelial cells. So during inflammation, if this site is going to have inflammation, P-selectin and E-selectin are induced. Then they bind Sialyl-X modified glycoprotein. I'll come back to this in the next slide. I got two slides mixed, reversed. So by recognizing glycan, this leukocyte got attracted to the site. And this is a low affinity interaction. So this leukocyte will roll on this endothelial cell, on the endothelium surface. But then as the inflammation signal continue to evolve, there's other ligands, other biomolecules expressed or induced. For example, ICAM1, and this is ligand for integrin. So the integrin on leukocytes will bind to this ligand, and this is firm adhesion. So now the cell was kind of stopped there, and then later on traverse through the gap created between endothelial cells, and they get into the site of inflammation. So after selectin, discovery of selectin was really big news in scientific research, and also also started to get glycobiology recognized by the scientific community. Then the next important progress was the discovery of the glycoligand recognized by selectants, and that is Silu-X, this tetrasaccharide. Then the scientists move on to try to find a receptor for selectin, and for the first one that was discovered was PSGO1, so P-selectin glycoprotein ligand 1. And it turned out these are recognized by all three selectants. And later on, there are more and more glycoprotein ligands discovered that bind to selectants. So actually, this is the point I made earlier in terms of there's no one-to-one relationship between glycan-binding proteins and its glycoprotein counterpart, because unlike protein cytokine and cytokine receptor interaction, right? So actually, I believe in the selectin field, people do not talk much about what they bind to. They mainly study their function. This issue will become more obvious when I talk about galectin. Nevertheless, the field of selectin continue to evolve, and almost 20 years later, I think this is the first drug targeting selectin. It's a P-selectin. So these are two professors from University of Oklahoma funded this company, and they founded it because they study P-selectin, and they have antibody against P-selectin, monoclonal antibodies, and I believe this will be useful to maybe has post-therapeutic potential. That was 2002. Almost 10 years passed, and they actually started to work on something they never expected they would get into, that is sickle cell disease. And they came up with the notion that the P-selectin may be involved in mediating adhesion of sickle erythrocyte to the blood vessel surface. So indeed, clinical trial using this monoclonal antibody, they show that the blockade of P-selectin by the monoclonal antibodies they have reduced sickle cell related pain crisis, or this called the vaso-occlusive crisis. This really very patients very much suffer from this disease. They almost have to go to emergency room each time there's a pain crisis. And the company was acquired by Novartis in 2016. And then this drug was approved by US FDA in November 2019. So 17 years after the company was funded. And this is actually a success story. I know the founders well, especially one of them, Richard Cumming. He later on moved to Emory and moved on to Harvard. He's now a program director at Harvard, a very successful glycoscientist. Siglec. So Siglec is sialic acid binding immunoglobulin-like lectins. So SI is sialic acid. Sialic acid is a monosaccharide, has 9 carbon instead of 6 of the other monosaccharide. So IG is immunoglobulin, and LAC is lectin. So Siglec. It contains one domain that binds to sialic acid, and the other immunoglobulin-like domain can be 2, 3, or many. I will come back to that, talk about that a little bit later. So in the cytoplasm, there's an immunoreceptor tyrosine-based inhibitory motifs, or ITIM. You know, there are ITAM and ITIM. ITAM is activating motif, and ITIM is the inhibitory motif in the cytoplasmic cells. So this implies that these proteins or glycoproteins play the suppressive role. They downregulate intracellular signaling pathways. There are 15, but in fact, the 16 is Siglec now. There are roughly 2 groups. One is conserved, meaning that if you want to study the mouse Siglec, you can find the mouse Siglec corresponding exactly to the human Siglec. They are CD33 related Siglecs. They are not conserved, and actually they evolve very quickly to the point that when you study one Siglec from in mice, you might not know exactly which Siglec in human it corresponds to. This caused some challenge to this field, because when you make mice without giving Siglec, which is a very common approach these days, you do not know the phenotype you observed tells you about what the function of the given human Siglec. Also note that this red circle ITIM motif. So most of Siglecs contain this ITIM motif. Siglecs are studied mostly in hematopoietic cells, leukocytes, and each leukocyte express one, two, or many Siglecs. And as I mentioned, that they are suppressive. So, for example, if this is B cells, when the activatory receptors, for example, this could be base cell receptor, immunoglobulin, when they are engaged, they mediate activation signal, or they transmit. The activation signal get transmitted. But if a Siglec, in this case a Siglec 3 or CD33 is also engaged, then somehow they will recruit phosphatase, and phosphatase is known to cleave phosphate in this in the signaling pathway, molecules in the signaling pathway, and leading to suppression or inhibiting or dampen this cell activation process. Just share with you one recent, very interesting, exciting development in the field of Siglecs. There are many examples. I'm sorry, I have time to talk about just one. So NK cell are known to express Siglec 7, and tumor cells known to express salicylic acid carrying glycoproteins. And in this case, these authors show that some glycoproteins on tumors can bind to Siglec 7. Then, because Siglec 7, like many other Siglecs, are immunosuppressive molecule, and so the NK cell function of NK cell is a natural killer cell. They can kill tumor cells. So interaction of salicylic acid ligands on tumor cells, they shouldn't be tumors. Should be tumor cells on Siglec NK cells may dampen NK cell activation in a tumor context. So I think you're familiar with PD1 or CTL4. That's the Nobel Prize 2 years ago. PD1 both are immune checkpoint, and immune checkpoint blockade now is the mainstream of therapy, cancer therapy. But we also know that they only a subset of given cancer can be treated or can be cured by this approach. There are other immune checkpoint molecule, and the blockade of those can maybe used together or maybe separately for treating cancer. This is a new direction in cancer therapy or cancer immunotherapy. And this Siglec 7 blockade might be one way. And there are other Siglec being discovered to have this immune checkpoint-like function. And I think I'm going to skip this one, just say that another therapeutic potential, this was published 2013, and I'm not sure this has become, whether the investigators are continuing to pursue this possibility or not. They generate this liposome containing actually antigens recognized by IgM on the B cell surface, and they also have molecules that can bind to CD22, which is Siglec 2, and the Siglec 10 or G, and then cause the suppression of B cell function and antibody production. So remainder time I will talk about galactans. 半乳糖凝集素。So galactans are beta galactoside binding animal lactans. So gal is a galactan, galactose, and lactan. So galactose binding animal lactans with conserved CRD, carbohydrate recognition domain. They evolutionarily highly conserved, a little bit different from Siglec. Siglec evolved quickly to the point that so mouse Siglec can look very different from human Siglec, although they are still share homology. They are found in nematodes, so very commonly used model organisms is C. elegans is nematode. They express quite a few of galactan family members, and all mammals express various galactans. Galactans can be one CRD. They exist as a homodimer, or two CRD in tandem, and this is separated by a short peptide linker. And galactans, we actually were the first to. My lab is the first to clone and sequence galactan 3. Back then, this is the galactan family was not known, did not exist when we first cloned and sequenced the protein later on known to be galactan 3. I'll talk about the 7 mainly and galactan 3 and maybe 8, and very quickly on 9 and 12. So galactans, in addition to binding galactose, they also share some signature sequence or consensus sequence highlighted here, and these are invariably present in among all galactans. However, the sequence identity among different galactans are actually not that high. So, for example, between 1 and 3, 1 and this part of 3 maybe just about 40% or less. And but between human galactan 3 and the mouse galactan 3, human galactan 1 and mouse galactan 1, there's maybe 80%, 90% identity. So it's highly conserved. For that reason, when there are so many members, one commonly asked question is whether there's redundancy. But I don't believe there are redundancy. Maybe between 1 and 2, but the others are very different among each other. So some galactans have wide tissue distribution. For example, galactan 3, I will talk about expressing many different tissues and cells. Others, like galactan 7, I will talk about, and especially galactan 12, they have a higher tissue specificity. I mentioned that the galactans are not transmembrane protein. They don't have transmembrane domain. They do not contain, or they are not synthesized with classical signal sequence or leader sequence. So they are not made through the Golgi pathway and exocytosed, get outside the cell. But consistent with that, they are found mainly in the cytosol. They can go to nucleus, but also in the extracellular space. We do not know exactly how they are secreted because they do not have classical signal sequence. Like many other lactans, 凝集素. So galactans are bivalent or multivalent with regard to their carbohydrate binding activity. So they can cause aggregation of cell glycoprotein. They can cause aggregation of cells, although we are not sure aggregation of cells of the function of this protein inside the body. For example, this galactan can cause hemagglutination. So this is a very convenient way to test the activity of galactans if you purify it from as a recombinant protein or from cell line. But this does not mean the hemagglutination is the activity of one of the function of galactans. So you might find that this field is somewhat complicated, but a good thing is they are getting more and more attention because they are implicated in the pathogenesis of a variety of diseases. This is mainly in recent years because now many galactans have, scientists have developed mice that lack a given glycan. So they are more and more knockout mice. And from there, by studying the phenotype, they have been implicated in the pathogenesis. So this is the something I wanted to share with you. I mentioned that the question mark following the extracellular function of glycans. So a variety of cellular responses can be induced in vitro by using glycans added exogenously to various cell types, because this is a multifunctional protein that bind glycan. So they can envision that they can cause aggregation of or cross-linking cell surface glycoprotein. And usually this means transmit signal transduction, right? And so this is easily understandable. Then if you go one step further, and indeed the researchers do that, have done that, that is, you inject glycans into experimental animals, then you find some response. However, and to mention that these may not represent the function of endogenous glycans. Issues, one is, first, the easy one is glycans might not be secreted or secreted in sufficient amount. So they might require a larger amount to induce activation of cells or cause response of cells, but they may not be secreted in the first place, or they may not secrete in sufficient amount. Then specificity is the point I mentioned. So unlike cytokine, has unique cytokine receptor, the one-to-one relationship. But the glycans, they do not have one-to-one relationship with a given glycan. So from the in vitro study mentioned on the top, we really cannot predict what glycoproteins on cell surfaces or extracellular matrices glycan bind to. If it's secreted in sufficient amount, we cannot predict where these glycans will go to or which cells they will bind to. I hope this is clear. I know this might be a little bit complicated, and over the years I try to convey this message. I'm not sure I've been successful, although I think it shouldn't be a too difficult concept. So for many years now, we actually used to just add glycan 3 to cells to see what happened, and conclude the function that way, and publish paper. In fact, I regret that we did that. But then subsequently, for many years now, we want to ask the function of endogenous glycans. Endogenous 内源性 does not mean it's intracellular, but it's just the cells come with these glycans. We want to ask what the function of those glycans they come with the cell, rather than what happened when you add recombinant glycans or purified glycan to cells. Then we ask whether, because glycans are mainly intracellular protein, whether they have intracellular functions. So we actually have been lucky that we succeeded in identifying a number of function of endogenous glycans, and these are intracellular function. Although each one took quite a few years. So the first one, this is the first intracellular function of glycan, is the anti-apoptosis. Anti-apoptosis, this is in T cell. And in that work, we showed also glycan 3 bind to very well known anti-apoptosis protein, BCL2. So we proposed that glycan 3 exert anti-apoptosis function by binding to BCL2. And the next one, this is very interesting, and I'm always very pleased with how we came up with this, the findings, that glycan firstly promote phagocytosis. This is in macrophage. And then when phagosome forms, when the phagocytes or macrophage engulf certain particles of cells, and then this substrate became a part of phagocytes. Then glycan-3 recruited to surround these phagocytes. Phagosome is on the cytosolic side. Then later on, we found in T cells, glycan-3 also translocate to cytosolic side of immunological synapse, and this is involve T cell receptor, and through T cell receptor engagement, the signal can be transduced, and glycan-3 can modulate that signal. And then we implicated ALEX that we found to be a glycan-3 binding partner. Through binding to ALEX, glycan-3 modulate T cell receptor mediated signal transduction. And then there are other findings later. We published. In fact, there are maybe 3 or 4 more. And so here, I should okay. So something is blocking. Okay, yeah, maybe this helps. Interaction with the intracellular interaction with intracellular regulators. So these are intracellular function, and inside the cell I mentioned earlier. Although they are glycoprotein, glycolipid, they are in the ER and Golgi on their way being synthesized, and then get outside the cells. But in the cytosol, there's actually, other than that old gluconeck I mentioned earlier, there's no glyco conjugates or glycoprotein, glycolipid in the cytosol. Okay. I should mention that the glycans do not bind to O-linked N-acetylglucosamine, which I talked about earlier. And this is so this binding is independent of glycan binding. So for example, BCL2 and the ALEX, they are not glycoproteins, but glycan-3 interact with them. However, the glycan carbohydrate binding site domain may be involved because this is a small molecule. I should have mentioned that each CRD is maybe about 120, 130 amino acid, and the carbohydrate binding site can be used for binding to protein, which is not too surprising. Okay. So then we have so many of this work I mentioned was we used the cells from glycan-3 knockout mice we generated, and we also studied the phenotype of glycan-3 knockout mice mostly in allergic skin and airway inflammation, atopic dermatitis, contact dermatitis, and asthma. And all these 3, we found that glycan-3 knockout mice have reduced or lower responses, and the pathways that involved the glycan-3 knockout mice have a lower response. So glycan-3 in this in vivo experiment, we show that glycan-3 is critical for T cell mediated allergic skin and airway inflammatory responses. There are many other papers published by other investigators, and also we have published a few others as well. So in general, glycan-3 is a positive regulator of a variety of inflammatory diseases. Although there are exceptions, they may be pro-inflammatory. Maybe they are inhibitory, but largely, mostly, this is positive regulator. Very interesting that glycan-3 now is well known to contribute to chronic heart failure. So this was first discovered by glycan-3 being a marker, actually in the blood, that correlate with the prognosis in patients with chronic heart failure. So actually, FDA approved the use of ELISA kit to measure the glycan-3 level, and then a lot of physicians or cardiologists to predict the prognosis of the patient, and then that might guide the treatment. In fact, I just did a search for keywords heart failure and the glycan just a couple of days ago, and there are already over 600 papers. So this, I believe, in the cardiology field, this is pretty well recognized. And there are a couple, but however, I found that mechanism is not known until recently. So these are two papers talk about Glycan-3 contribute to inflammation and fibrosis. These are both mouse models, and they use Glycan-3 knockout mice. However, I mentioned that the Glycan-3 is first found to be serum biomarkers. So many investigators would think that they are some kind of mediators. They could be function like cytokine and to lead to inflammation, fibrosis. However, that might not be the case, and that's what I mentioned about the extracellular function. And so these two papers actually did not address whether extracellular, but mainly talk about the phenotype of mice. So this is still work to be done, and this is important because if you want to find develop inhibitors and for treatment of heart failure, you need to know where Glycan-3 function. And similarly, in renal diseases, there are papers already published about Glycan-3 contribute to fibrosis. So I found nice review articles on Glycan-3 in autoimmune and non-autoimmune nephropathies. I need to mention that when you read a review like this, in fact, include the review we published earlier, some of the conclusions made might be related to the extracellular function that published earlier. So you need to kind of dissect what is the information contained in the review in terms of which one is result from the study of endogenous glycan, which one is study of exogenously added glycans. So from all this research, Glycan-3 may be targeted for treatment of inflammatory disease. That's related to the title of today's presentation, the therapeutic potential. But then I ask, should we target intracellular or extracellular glycans? Okay. So I think you probably guess what my answer is. Actually, there are several companies working on Glycan-3 inhibitors or developing therapeutic based on inhibiting Glycan-3. The most successful one is Glycan-binding. This is the company established by a professor from Lund, LUND University. They are a chemist and biochemist. They started this company. Started with making inhibitors for Glycan-3. The TD139 here is now very well recognized, very well known. In fact, it became commercially available. It's a derivative of TDG, that is a thiol diglycoside. And the company has succeeded in showing in phase 1, and I believe it now in phase 2, that the TD139 was effective in treating very difficult disease to treat, that is idiopathic pulmonary fibrosis. So I talked with the company, and they are not quite sure how TD139 work in blocking Glycan-3 function. But we know, and they also know, that TD139 is cell permeable. So it can get inside the cell and block Glycan-3. If Glycan-3 is for the pulmonary fibrosis, Glycan-3 working inside the cell, then it can work as well. If the company is started by immunologists, then they probably will be just like the other company on Selectin. If they probably would have started with anti-Glycan-3 monoclonal antibody, then I wonder whether it will work or not. But this is just my biased opinion. Okay. So quickly on Glycan-7 and psoriasis, mostly is our work. And psoriasis, I think you are familiar with, so I won't read this in the slide, but just say this is another inflammatory disease, and there's a hyperproliferation of keratinocytes, epidermis, and also inflammatory infiltrate. And I hope you know Th1, Th2, Th17. This is mainly Th17. Also, I want to emphasize this is a metabolic syndrome, meaning that the patients are usually obese and they have dyslipidemia. I will make come back to tell you why it's relevant. So this clinical slides, a patient can have over 80% or maybe full body covered by psoriatic lesion, and this is a very typical psoriasis plaque, psoriatic plaque. And you don't need to do biopsy to recognize this, if you're especially dermatologist. But if you do biopsy, you find this so-called psoriatic form hyperplasia. And then if you stain for leukocytes, there's a dense cell infiltrates, both in dermis and epidermis. So Th17, meaning that dendritic cells are known to drive T cell into Th1, Th2, Th17 direction polarization, and depends on the cytokine they produce. So if it's IL-23, they drive to the Th17 direction. And either Th1 or Th17 cytokines can then cause keratinocyte to be activated and proliferate, and then subsequently this will lead to generation of psoriatic plaques, and because there's cell proliferation and also increased inflammation, because keratinocyte can actually mediate the inflammatory response. And this is another way to look at psoriasis. It's a little bit out of date, but in general, we know that dendritic cells play an important role, and through the Th1, Th17, mainly Th17, T cells and cytokines, keratinocytes get proliferated, activated, and they can in turn attract and activate neutrophils, and all come back to they come back to activate dendritic cells. So this is the amplification process here. And so Glycan-7 is expressed in stratified epithelial cells like skin keratinocytes or esophagus. They're highly induced by p57. Glycan-3 is the anti-apoptotic. Glycan-7 we show that it's pro-apoptotic and it's tumor suppressor. Another group showed that Glycan-7 is bind to BCL2. We show Glycan-3 bind to BCL2, but Glycan-7 also. The right side is mainly just to impress you that try to impress you that this intracellular protein and also can be highly enriched in nuclei. So we work on psoriasis. The work is mainly done by two postdoc fellow, Chen Hongling and Luo Jiahui. The reason we got interested in Glycan-7 is we conducted a transcriptomic analysis with Genentech, a biotech company, and the paper is already published. We look at atopic dermatitis, contact dermatitis, and psoriasis. For each one, we recruited 10 to 15 patients and the biopsy the non-lesional and lesional skin, and look at the gene expression profile. And for psoriasis, we found Glycan-7 is downregulated in the lesional skin, downregulated. And this was confirmed by immunohistochemistry. It's lesional, non-lesional, and less lesional. You see, it's less intense staining for Glycan-3. So our question was what happened to the how Glycan-7 downregulation through downregulation cause the psoriasis development psoriatic lesion through keratinocyte. And we have this already published. So we found Glycan-7 actually can regulate, modulate expression of actually only 4 microRNA. So microRNA 203, and it's upregulate microRNA 203, and this MIR 203 in turn suppress p63, and the p63 is known to cause keratinocyte proliferation. So then we Hongling mainly show that Glycan-7 can activate JNK1, C-jun N-terminal kinase. This is kinase, and then we concluded that the Glycan-7 through this pathway suppress keratinocyte proliferation. When it's downregulated, there's increase in proliferation. So this take care of the epidermal hyperplasia part. Then on the inflammation. So another microRNA that Glycan-3 regulate, in this case is suppression. Glycan-3 suppress microRNA 146A, And we show that MIR-146A is a promoter of this MAP kinase pathway induced by IL-17 through IL-17 receptor, leading to IL-6, IL-8 production. These are two major cytokines contributing to psoriatic inflammation. So Glycan-7 suppress these pathways by suppressing microRNA 146A. So we were able to conclude from two papers, downregulation of Glycan-7 in keratinocytes may contribute to the heightened keratinocyte proliferation and the inflammatory response in psoriasis. Thus, approach to induce Glycan-7 in skin may be developed as a new therapies for psoriasis. And we're lucky that by connectivity map analysis, this is CMAP. I'm not sure you're familiar with this, but that's okay. And it's very powerful in silico analysis. We found that statins, I'm sure you're familiar with statins, can induce Glycan-7 expression in keratinocytes. Thus, we propose that the statin may be employed for treatment of psoriasis. In the meantime, control this lipidemia. So we propose, we actually already proposed a clinical trial based on this, and statin alone, we don't think it will be effective enough, but together with other treatment, believe will benefit patients, not just for their psoriasis, but also for their dyslipidemia and for their metabolic issues. Okay, I will skip next few slides because I really want to get to the last part. So the first part of the slide I missed, I skipped is Glycan-9 and Glycan-12 is very interesting glycan that we discovered and has to do with lipid and it's expressed mainly in adipocytes. So I hope I will. Actually, I talked about this earlier during my earlier visit to Tzu. Maybe share with you about our recent development with regard to this glycan, maybe in the future. So, because I want to talk about this final part. So, come back to the extracellular function. Although it can be easily demonstrated, I continue to ask whether exactly what glycans do in vivo, also in extracellular space. And although we know that they are able to engage cell surface glycoprotein, I gave you a number of examples of Glycan-3, 7, and I skipped the Glycan-9, 12. Inside the cell, they also can have an important role. They function by glycan independent, in a glycan independent fashion. So we've been asking to cytosolic glycans function by binding to glycans. So by glycan, I meant this slide I showed very early. So O-glycan, N-glycan, glycolipid, and proteoglycans. Inside the cell, I mentioned that they are all linked N-acetylglucosamine, but the glycans do not bind to these glycans. And the answer is glycans really not supposed to bind these glycans because this glycoprotein, glycolipid, the synthesis is initiated in the lumen of ER and the Golgi ER, and then they get modified in Golgi, and they can get outside the cell. They can be internalized and get into endosome and be digested, or they can recycle back to cell surface. But all this time, these glycoconjugates, glycoprotein, glycolipid, in the lumen, of these organelles, ER, Golgi, post-Golgi vesicles, and endosomes, lysosomes. However, we didn't know. I wish I knew this earlier. I very much regret I didn't know about this actually can occur. So that is, they are disruptive materials that can disrupt endosomes or lysosomes, maybe other organelles. So this disrupt material, I've come back, they are various kinds. They can be internalized, and this is in the process of endocytosis. In endocytosis, the glycoproteins, glycolipid, initially on the cell surface can be internalized, but they stay in the lumen, just like I mentioned in an earlier diagram. They are in the lumen of these vesicles, and glycans are outside. However, this disrupted material can cause damage to these vesicles, and then the glycoprotein, glycolipid can be exposed to cytosol. In cytosol, there are glycans like glycan 3, glycan 8. They can bind to these glycoconjugates very quickly, very readily. And then in addition to binding, it's now known that they can induce downstream events, for example, autophagy. Yeah, this is a very interesting phenomenon or biological process. And so we were studying the infection of macrophages by Listeria. This is a very well-known intracellular pathogen, and they can be engulfed by macrophages, or they can invade the macrophage. But they are one kind of a disrupt material because they want to escape from phagosome. They cause damage to the phagosome. They can get outside the cells. So we show that glycans, this is time lapse, and glycan 3 can accumulate it quickly around. So this lysis process occurs about 1 hour or so, and just around 1 hour, glycan 3 can be accumulated quickly around where Listeria was. Okay, this very important papers published. Very important paper published by a German group. That glycan 8 targets damaged vesicle for autophagy to defend the cells against bacterial invasion. So they work with Salmonella, and we work with Listeria. And the Salmonella, like Listeria, can be engulfed or can be phagocytosed, but they want to escape and cause damage to the phagosomes. Then these authors published this paper published in Nature. This is 2012. There's already maybe close to 900 citations. I believe this will become the most highly cited papers in glycan field. So they show that 3, 8, and 9 can bind to the glycan in damaged phagosomes, and more importantly, as importantly, glycan 8 can recruit autophagy adapter NDP52 and induce autophagic activation. So we had to catch up, but very much inspired by the work, and then showed glycan 3 can function oppositely. That is, glycan 3 suppress this autophagic activation. So we published a paper a few years later, and this became editor's choice. Glycan 3 protects intracellular Listeria by suppressing the autophagic activation or the autophagy response in a host and glycan dependent manner. So this is glycan 3 bind to host and glycan, and 3 and 8 antagonize each other. Another example is with H. pylori infection, but I need to only go over to the final conclusion slide. So H. pylori, which you are familiar with, cause peptic ulcer. They are not intracellular bacteria, but they can generate produce virulent factor, for example, VAC-A. So vacuolating cytotoxin A cause damage inside the cell, and we show that VAC-A can cause lysosome to be damaged. Then glycan 8, just like in the case of Listeria Salmonella, can induce bind to the to damaged lysosome, bind to glycans in the damaged lysosome and induce autophagy. And we show further that the autophagic activation can lead to generation of reactive oxygen species and cause additional damage of lysosome. So these are important because there are many substances that cause endolysosome damage. Intracellular pathogen or pathogen that produce core-forming proteins like VAC-A. Virus actually can also cause damage to endosome. Protein aggregates like those involved in neurological disorders. So my colleague Chen Yizhuang 陈一川老师she in collaboration with us show that in the disease she's expert of Huntington disease. The problem of the disease, the causing substance is the disease causing substance is mutant Huntington. These aggregates. Then they show these aggregates can cause damage to lysosome, and then this is known to cause inflammasome activation and Glycan-3 potentiate this process through binding to damage lysosome, and this causes increased inflammation in microglia in the brain and cause neurodegeneration. So this is the protein aggregates, but also particulates. There's not many examples, but we believe that, for example, PM 2.5, PM 10 particulate matters, and in fact, the nanoparticles that you're now familiar with because of the vaccines, they can cause this kind of process and leading to some inflammatory response, which is I'm not sure is widely recognized. And finally, cellular stress, that the oxidative stress is predicted to cause endosome lysosome damage. So so I'm sharing with you a novel mechanism mediated by cytosolically exposed glycans. And this can lead to diverse cellular responses, autophagy, inflammasome activation, maybe apoptosis, and this can lead to innate immunity in the case of infection, inflammatory responses, and this is associated with diseases. So I then come back to this slide. So outside the cell, we still do not know exactly what they do. They must be doing something important, but inside the cell, I share with you, they can function in glycan independent fashion, but also very exciting new recent development is they can engage intracellular glycan dependent glycan sensing responses. So I think I'm already exceeding almost. Actually, yeah, my time is up. So again, thank you for your invitation and thank you for your time.
Just raise your hand and ask. Anyone want to share your opinion? Maybe I have a question for Professor Liu. Because you're talking about the Gelatin-3 with the Gelatin-8, and more like the antagonize each other in some tissue or cell. Is that for every kinds of the cell or tissues? Because it's very interesting.
Yeah. So yeah, very very good question. So I must first clarify. We look at Listeria, and then the other group look at Salmonella, and we didn't look at Gelatin-8 in relationship to Salmonella in our system. So, but we believe also from other studies, this Gelatin-8 is in promoting autophagic activation. Gelatin-3 in other systems as well is suppress autophagic activation. So I believe this can be generalized in this endosome lysosome damage process. So yes, I think this can maybe generalizable, which is what something we were interested in doing, very interesting in the first place. I should mention that they bind Glycan-3 and 8 bind to different set of glycans. Maybe there's some overlap, but they bind different set, and they do not inhibit each other in terms of binding to glycan. So it must be in the autophagic activation process. Could be so Glycan-3 is not known to interact with NDP52. So it's through other mechanisms it suppresses autophagy. Glycan-8 promote autophagy through binding to NDP52. Yeah.
Okay, thanks. Is that the Chen?
Yeah.
Raise your hand. Okay.
Fudong.
Yeah.
I have a question. So very similar to the previous question. So actually you just mentioned that Glycan-3 and 8, they do not interfere their binding to glycan.
Right.
Assuming that when you have
not antagonize each other.
Okay. So when you have a damaged endosome, that so both of them will bind to the damaged endosome and expose glycan at the same time.
Yes, right.
Oh, okay.
Right. However, but through a super resolution SIM, we found they bind to different microdomain.
I see.
So
I
it's
see.
really very interesting. So
it's the later part how they engage with autophagy.
Right. Yeah.
That differentiate their function later.
Right. Yeah.
Okay.
We still have quite a bit to learn there. Yeah.
So did your team find out that like different stimulation, I mean different damage, induced endosome damage will lead to different accumulation? Like in some cases you will have more Glycan-3, and in some cases you have more Glycan-A, or normally you will find equal amount.
So yes, not exactly to that extent, but we use the photosensitizer and then illuminate. This is the system developed by Yang Weiyuan, and so we modify a little bit. So we cause damage to endosome. He caused damage to lysosome. So the endosome, then we find the 3 and 8 really behave very differently. And so that's another system. We hope to answer your question more in more detail is H. pylori. So the study was Glycan-8, but we also have Glycan-3. Already have quite a bit of data. Have not published yet, but there I hope to compare 3 and 8, and answer the question you addressed. And again, also see whether they antagonize each other or not, and also the timing of their binding to the glycan. I should mention in the photosensitizer case, Glycan-8 bind very, very quickly, like within a second.
Oh,
we shine a light and it's there. And 3 take about a minute or so.
I see. Okay.
Yeah.
Oh, okay. Thank you.
Yeah. Thank you for the question.
Professor, I have a question.
Oh, okay. Yeah. Just ask.
Okay.
Maybe just introduce yourself a little bit.
Okay. Professor, my name is Ayo. I'm a PhD student, year 2.
Hi.
In Sydney.
Hi.
Okay, sir.
Yes.
So my question is this. First, I want to ask about how to solve the problem of the specific binding of the glycan-binding proteins, because since there is a problem of them binding to different kinds of ligands. So how do we circumvent this event? That's my first question.
Yeah.
Like, how do we actually ensure that this does not happen when we begin to investigate any of the glycoproteins?
Can I answer this first?
Okay,
so
sir.
Very, very important question. I'm very, very happy that you asked this question. So I think it's 2 aspects. One is we just need to be aware of this issue. And for example, in the literature, including what we published earlier, it's very common that one of the glycans is linked to one glycoprotein. Then the rest of the paper or rest of series paper is focusing on that interaction, and which more and more clear is that may not be the case. So typical example is Glycan X bind to a CD something, and then show that there's a response. And then conclusion is through binding to CD something, Glycan X induced that signal, right? And then which is then later on find that this may not be the case. So just first be aware that we call the almost like a multivariant kind of situation, aware. And then the second aspect is maybe these glycan binding proteins are really different from the cytokines of those. Their function might just involve binding to many partners at the same time. For example, intracellularly, I talk about this damaged endosome. I didn't say that which glycoprotein they bind to. I believe they bind to quite a few. The Glycan-8 bind to a set, but Glycan-3 bind to another set, and they don't. They actually it's totally separate. But in that set, there might be more than one glycoprotein. So some cases, you really do not need to know the specificity. Mainly glycans as a group are suited to serve as anchor, and for glycan to engage glycan, right? So that way we don't need to solve, just need to be aware. Also need to be aware is extracellularly. Then what you need to solve is extracellularly. So what are you going to do? I believe it's very challenging. That's why after so many papers, I still put a question mark there. That outside cell, we need to know, we need to work with animal models and find out, say, one glycan coming out of macrophages, where they go to. They go to neighboring endothelial cells or epithelial cells or T cells, and then in terms of what cells get activated. So this really requires some kind of maybe new tools or the combination of many tools to dissect this, right? You see what I'm saying? Yeah, so we need to really go in vivo, in animal, and then look where glycans are. Might not need to know which glycoconjugates, glycoprotein bind to, but mainly figure out where they go to. That's how I see that as a first step. That is already challenging now. And then which cells are activated. Then we can say, yes, they're doing something outside the cell. Yeah. The mere presence of these glycans may not be the answer. So you find it in the serum does not mean it work outside the cell. Yeah. I hope it's clear. I'm sorry. So your next question?
Okay, thank you, sir. My next question is, what role does, maybe because you've said that, okay, the gelatins, they play intermittent roles together, but in ubiquitination, which is also a process of maybe degrading proteins as well, have you worked on anything that has to do with that? Maybe is there an interaction or a link with that?
We don't work with that directly, but other researchers have published paper also has to do with endosome damage or lysosome damage, and they start to put the ubiquitination there, and in a kind of a pretty complicated scheme. So I think all these different processes probably interwave. Eventually we need to sort those out as well. So no, we have not looked at that directly, but the other researchers are looking at ubiquitination also, because it's also can maybe more ubiquitination also occurs when proteins are degraded in lysosome. So these are clearly somehow related, but we have not addressed that directly yet.
Okay, sir. Then my last question has to do with autophagy, because I'm interested in autophagy. I understand that for autophagy flux, it means that like autophagy process has been completed. So for the galectin 3 and 8, because you said they don't necessarily inhibit each other, does it mean that do they enhance autophagy flux, or like for galectin 3, does it suppress it or enhance it? Because one of the paper that you mentioned, I think there is like 2 different roles between the 3 of them. So right. Yeah, I just want to be clear, maybe which one favors autophagy flux, or does it have to do maybe in a disease condition? I mean, does it relate to that would determine maybe autophagy is actually activated or is downregulated?
Yeah, so it's really still we don't know. We don't have answer to all those. It's still pretty early in this area of research. But mainly what we know is galectin 8. Only among 8, 3, 8, and 9, only 8 engage NDP52. So it's generally accepted that when NDP52 is engaged, then there's autophagy activation. So that's all we know. And the galectin 3 does not bind to NDP52. So through what mechanism is suppressed autophagy, we do not know. We cannot know. So still lots of work need to be done. Yeah. Yeah, very good question. Thank you.
Okay, have anybody have do you have a question? No? Okay. So maybe we just have a break because I think the Professor Liu just give us a wonderful talk for 1 hour. So just give the warm hand for the Professor Liu, then we will have the break. Okay. Thank you, Professor Liu.
Thank you.